Thank you all for joining us at the cutting edge of cardiovascular sciences. I'm John Cooke. I'm professor and chair of the Department of Cardiovascular Sciences here at Houston Methodist Research Institute and Hospital. And joining me in the studio is my colleague and guest, Dr. Ashrith Guha, who's going to be talking to us in a moment. He's a cardiologist with particular expertise in pulmonary hypertension and right heart failure. But first, we'll be talking to our guest joining us by Zoom, Dr. Harry Karmouty-Quintana. And let me introduce Harry, Dr. Karmouty-Quintana, to you. Dr. Karmouty-Quintana got his PhD in pharmacology and biophysics from King's College London. And there he was one of the first to apply MR to studying lung disease. Subsequently, he went and did a postdoctoral fellowship at McGill University, which is a very famous medical and research university. Then he came to Texas, and at UT Health he joined as a postdoc fellow in 2010 and continued there as now an assistant professor at UT Health and doing some amazing work in pulmonary hypertension. And today we'll be hearing about a new form of RNA regulation that he's studying in that disease of alternative polyadenylation. Dr. Karmouty-Quintana, thank you for joining us.
Thank you. It's a pleasure to be here. Thanks, John, for the introduction. So I got into the field of pulmonary hypertension because I was interested in joining UT Health. One of the biggest problems there was we identified what patients that had lung fibrosis, which IPF, many of them developed pulmonary hypertension. So we were very interested in understanding the mechanisms that led to this. And through this research and also through all this interest and through working with other labs in the biochemistry department, we came across this concept of alternative polyadenylation that at that time was really linked with proliferation of glioblastoma. So that was one of the first publications that really put alternative polyadenylation in the map. So that's how I got into pulmonary hypertension. So it's okay with you, I'm going to start now with the presentation.
Sure, why don't you go ahead and start.
Right, so this worked earlier, but now I'm having trouble finding the presentation.
Okay, that's fine. While you're getting the Zoom up there, let me just introduce Dr. Ashrith Guha while we're waiting. Dr. Guha is a cardiologist. He specializes in right heart failure, pulmonary hypertension, cardiac transplant, and received his medical degree in India, and then moved to University of Alabama, where he completed his master's in public health, specializing in epidemiology. He did a residency at University of Texas, and then he stayed on as an assistant professor, and before he went off to University of Iowa to get his advanced heart fellowship training. And then fortunately he came back here at Houston Methodist Hospital to do his faculty position, where he is now associate professor?
Assistant professor.
Assistant professor, soon to be associate professor in cardiovascular medicine. Thank you for joining us, Ash.
Thank you very much for the invite. This is a great forum, and look forward to presenting our work.
So, Dr. Karmouty-Quintana is going to be talking about pulmonary hypertension and this alternative polyadenylation, which seems to be playing a role in the pathophysiology of heart failure.
As well, yeah.
Well, when I had this before, I could select PowerPoint and share it. Now I don't see it.
Well, you know what we could do, Dr. Karmouty-Quintana, we could go to Dr. Guha's slides if you want to do that.
Sure. I think
while we're waiting.
So while we are waiting, I just want to briefly introduce the audience to pulmonary hypertension as a disease process. So. Pulmonary hypertension is a condition where there is excessive, remodeling of the blood vessels of the in the lung, mostly the small blood vessels, the pulmonary arterioles, and that leads to the development of pulmonary hypertension. Normal blood pressure in the pulmonary arteries is about 20/10 millimeters of mercury, and in a disease state it increases to as high as 80 or 30, and the resistance normally should be about less than 3 Woods units or 240 dynes per centimeter square, and less than that, and in the disease state it increases. So as that increases, it puts a great stress on the right ventricle, and the patients who present with pulmonary hypertension mostly present to us with right ventricular failure, because that's the downstream ventricle which sees the pressure, and eventually all the patients who progress to death or lung transplantation because of pulmonary hypertension being progressive disease, mostly do so due to RV failure. That's why for us right ventricle is so important, especially as a heart failure specialist.
Could we talk for a minute about how the pulmonary hypertension itself develops? The end consequence is right heart failure.
Right.
Over time, though, it takes some time for the pulmonary vasculature generally to develop an increase in resistance that results in right heart failure, unless it's a pulmonary embolism, which is very rapid.
Right. So usually, there is an insult, and whether that insult is hypoxia, or initially it was a lot of these weight loss medications, which really, fen-phen was what really drove the epidemic of pulmonary hypertension in the 80s.
Fen-phen, remind us what that referred to.
Fen-phen is a weight loss medication, which is it's phentermine, which is very similar to the amphetamines. And initially there is vascular injury. The endothelial injury oftentimes is what heralds all the processes, which is intimal hyperplasia, smooth muscle hypertrophy, and also now we know that as all of those changes take place, there is small vessel thrombosis leading to sort of this vessel loss that we see, and then eventually, like you were saying, it takes probably a few years before it develops into clinically relevant pulmonary hypertension.
So it sounds like the process starts in many cases with an endotheliopathy, an alteration in the endothelium.
Exactly.
Yeah.
Exactly.
And then you lose control over vascular smooth muscle cell proliferation. You lose control over hemostasis, thrombosis.
Right. Right, and then now we also know that there is, the perivascular lymphocytes, or the lymphoid tissue, is also playing a big role in it, and that has been sort of some of the newer discoveries that have happened in the pathogenesis of pulmonary hypertension has been in the area of sort of the immune cells playing a role, and we know now that they play a big role, especially the macrophages. And also the T lymphocytes.
Marlene Rabinovich at Stanford has some nice data showing that there's an alteration in the endothelial to parasite interaction in pulmonary hypertension also, that you need that interaction between the parasites, the vascular smooth muscle cells that are wrapping around the endothelium. You need that interaction for the endothelial cells to remain viable. If you lose that interaction, the endothelial cells will die, and then you lose bits and pieces of your microvasculature.
Right.
That could increase pulmonary hypertension as well.
Absolutely, absolutely. So it is really a vascular disease, which I guess like you clearly explained, it's sort of a endotheliopathy. So,
well, why don't we go to your slides while
I think he has his slides available, maybe.
Okay. Is Dr. Karmouty-Quintana ready to start? Okay, we're waiting to hear about that.
Okay.
You can go ahead with your slides.
Okay, so wonderful. So I think we'll start off with this this first slide. I'll spend a few minutes on this one because I think it's important to understand what happens in pulmonary hypertension. So, like you were saying, so if you were to follow the heart from the, I guess, left to the right, where, the right ventricle is facing all that pressure, and initially when the right ventricle sees that load, what happens in the first phase is that to accommodate to the stress of the increased pressure, the right ventricle will become thickened to maintain the same volume and as to just essentially compensate for the stress, the increased stress that it's seeing. And that is the adaptive phase. So what you really need is what we call as the RV and pulmonary artery coupling. So the RV gets hypertrophied so that it's able to still contract and generate the amount of force that is required to push the blood into the pulmonary artery. So there is good coupling between the afterload and the contractility. And then eventually over time, that as the pressure continues to be sustained, that's when it becomes uncoupled, where the RV dilates and the walls start to thin out. And that is what we are really, interested in, because that's when patients present to us with symptoms. And so as long as you can keep the coupled state, and we see this often in adult congenital heart disease patients, who have had their disease very early in life, that the RV continues to be coupled for many, many, many years compared to a pulmonary hypertension patient who develops disease early, late in their life. The coupling, the RV becomes uncoupled very soon.
I got a question for you. So it's a decompensation of
Exactly.
the ventricle. The ventricle can no longer withstand or generate the contractility required to deal with the increased resistance in the
Right.
lung. Is it possible, Dr. Guha, that the vascular problem that is occurring in the pulmonary vasculature is now occurring in the right heart?
Oh, that's exactly what what the next slide will talk about. We think that, and that's the hypothesis here too. So let me go to the next slide here. So what as Dr. Harry will talk about later, and he's one of our collaborators that we work with. So you are absolutely right that there is vascular failure, which means the same kind of vascular issues that are going on in the lungs. Even though they're not exactly the same in the heart, but there is a problem with the vasculature where it's not just able to keep up with the demands of the myocardium, and which has been described before, and which we would like to also describe it as kind of vascular failure, which leads to progression of the right ventricle from a coupled or the compensated state to an uncoupled or a decompensated state. And our hypothesis was that we wanted to study if the alternative cleavage and polyadenylation of the mRNA actually plays a role in this vascular failure. So that was what we were exploring to study.
And we're going to be hearing a little bit more about alternative polyadenylation in just a moment from Dr. Harry.
Right.
But why don't you go ahead and keep rolling with this and tell us what you found.
So, we as a lab, because we do, I'm a heart transplant cardiologist as well, and we have a heart failure lab which has a large repository of hearts collected from explants, at the time of transplant. We also do a lot of, we are one of the largest heart double lung transplant programs in the country. We thankfully have the opportunity of having the hearts of patients who have had end-stage pulmonary hypertension. So what we did was, we took those hearts of the patients who were transplanted with end-stage pulmonary hypertension and looked at their RVs. And here, you can see that in patients who have RV failure, that there is increased fibrosis and increased hypertrophy in human RV failure compared to the controls on the left side.
It looks like the myocytes, the cells themselves, are larger in RV failure.
Exactly.
Is that correct?
Exactly. So there is definitely hypertrophy, and there is increased fibrosis.
Is that the thick strands of, well, it looks white in this microphotograph. Where are the strands of fibrosis?
Fibrosis would be.
That's fibrosis,
Right.
okay.
Right. And we wanted to also show that what has been studied mostly in the LV and some in the RV, that when you have RV failure, that you actually have essentially lack of microvasculature, or what we would call as vascular failure, which means for the density of the myocardium, then you have a lesser number of vessels than that would be needed to support that amount of myocardium, and we saw that in RV failure here. As you can see, the number of capillaries are much lower in RV failure. So we were able to show that yes, there is some vascular failure in
Can we just talk about that for just a moment?
Sure.
I think that is a fascinating slide, because on the left, on the upper panel, you're staining with isolectin before. That's my understanding is that binds to the endothelial cells,
Correct.
the isolectin,
Correct.
and probably has GFP or some green
fluorescent,
some green fluorescent protein. And then down below that, down below the two green slides, in the lower two panels, you've got CD31 staining. My understanding is that's an endothelial
staining as
antigen.
well. Yeah.
So in both cases, I think you can see, I think it's most easily seen with the isolectin staining.
Correct.
There's a dramatic reduction in the number of endothelial cells staining between the left panel at the up top and the right panel at the top. So the right is the microvasculature in RV failure, markedly reduced.
Correct. Correct.
That's fascinating. So there's a vascular problem.
Correct.
In the right heart that is failing.
Correct.
Interesting.
And then, we wanted to sort of look and see, as we wanted to study this APA, whether that is playing a role or not. So what APA does is just, I know Harry's going to talk about this, but what APA really does is it changes where the splicing happens. So there is a proximal site and there is a distal site, and what happens is, because of alternative polyadenylation, the splicing happens more at a proximal site, and then that leads to shorter mRNA transcripts, and because they escape regulation, you have enhanced translation, leading to more protein, and at least in the disease state. So what we did was...
So this alternative polyadenylation, APA you call it.
Correct,
APA.
correct.
So that basically it's changing the population of message RNA that the cell has.
Correct.
Changing the population of message RNA. Well, the message RNA encodes proteins.
Right.
And so if you're changing the form of the message RNA, you're probably changing the proteins that are...
Correct.
And that could play a role in disease. And
right.
So
So what we did was, we looked at, this is just the heat map of the RNA-seq that we did from RV and the controls, and saw that some of the pathways, which again we were interested in angiogenesis, and there were the integrins, and I'm sorry, it's a little bit smaller on the left, but TGF beta, a lot of the pathways which have been shown in other disease states to be altered in the endothelial cells. We saw that they were also altered here in the RV failure patients.
So I guess this is a heat map of RNA sequencing data that you have.
From patients with RV failure, and also with, compared to controls.
Compared to controls. So the left column are the controls, and the right column are the patients with
it's
RV failure?
other way. It's
okay.
Yeah.
But I guess what you saw is that there was a higher expression of inflammatory NF-kappa B signaling, inflammatory proteins, inflammatory genes, RNA-seq, and adrenergic signaling was also increased. So
right, which is expected with especially with any of the when ventricle hypertrophies and is failing, like we've seen on the left ventricle, that the adrenergic signaling is
upregulated.
Yeah. So
okay.
So here, what we do have collaborators in UTMB, Dr. Eric Wagner and his group, who have helped us with the other projects, especially with regard to pulmonary hypertension. So they have this specialized software to kind of see which, whether there is 3-UTR shortening and lengthening. So they were able to show that certain genes here are definitely shortened, and in the next slide you will see that there are multiple genes again which are lengthened as well. So this is essentially a gene enrichment analysis, and you can see the pathways to the left, and which are listed, and so we were able to show that yes, it does appear that this 3-UTR shortening is happening, APA is happening in the RV failure sample, the human sample, compared to controls.
Yeah. So 3-UTR shortening, the shortening of the 3' UTR, that can change the stability of the RNA construct. I guess it could change what microRNA is binding to it.
Exactly.
What RNA binding proteins are binding. That changes the regulation of the RNA.
So, and also the proteins translated. So then we sort of turned our attention to one of the regulators of this APA, which is NUD21. So there are about, I think, now 13 regulators of this APA machinery which have been identified, and one of the most studied, and what we had, along with Dr. Harry's group, had studied in pulmonary hypertension, was this NUD21. And we wanted to look at that and see what whether that was upregulated or downregulated in RV failure, to explain why we were seeing some of these, in the gene enrichment analysis. We did see that there was some 3-UTR shortening, so we had to see if any of the, the regulators of APA were upregulated, and yes, here it did look like NUD21 is actually higher in RV failure, compared to RV control.
Okay, so this is one of the proteins that regulate where the polyadenylation is occurring, is it?
Correct.
And it itself seems to be altered in right heart failure.
They're at least upregulated. Increased in.
It's upregulated.
Increased in.
Now, was its 3' UTR also shortened?
See, that of NUD21.
Yeah.
I don't know. We have not looked at it, but that's a good question.
Because that might increase its expression.
Correct.
Okay.
So then we went ahead and looked at, since our hypothesis was to look at the endothelial cells and look at vascular failure, so we wanted to look and see if, we wanted to stain the NUD21 positive and CD31 positive, which means those which have an upregulation of NUD21, and the CD31, of course, stains the endothelial cells. So we want to look at those endothelial cells which were positive for NUD21, and as you can see here, they were much higher in the RV failure group compared to, even though on the top bar graph you can see that actually the number of vessels are less, because NUD21 negative CD31 positive cells are actually less, which means kind of the endothelial cells are actually less, and we've seen it in the earlier slide too. Even though those are less, the proportion of those which are NUD21 positive and CD31 positive cells are much higher in RV failure, suggesting that maybe these NUD31 increase in these endothelial cells is playing a pathogenic role. Again, here we looked at this. So we went back, we have an animal model of monocrotaline, which is a very well-established model for pulmonary hypertension. So we actually looked to see if the same kind of signal we are seeing in the right ventricle from these monocrotaline rats, and we saw the same thing.
So monocrotaline is a drug that causes pulmonary hypertension? Okay.
That's correct. So in rats, and so we have that model in our lab, and we looked at the right ventricle from these rats and saw that again, you know, the NUD21 and CD31 positive cells are higher in these MCT rats compared to control.
Okay.
And then what we did was we looked at some of the public databases for other models of pulmonary hypertension. One of them is the rat model of pulmonary artery banding. One of the criticisms of monocrotaline is that it kind of, depending on the dose that you give, it leads to the systemic inflammation, including myocarditis. And there is always this question of how much of the damage was caused by monocrotaline itself, and how much of it is due to the pulmonary hypertension. So that's why we looked at this model. Again, we didn't do the experiment, but we looked at publicly available databases who have already published RNA-seq data from these pulmonary artery banding in right ventricle. And even here, if you can see on the left side, you can see that NUD21 is definitely increased. The expression of NUD21 is increased in the right ventricle of this pulmonary artery banding model. Again, suggesting that we are probably on the right track, saying that this NUD21 is definitely playing some role in these.
That's the cone on the far left there.
That is correct.
Indicating an increased expression of NUDT1
correct.
in the ventricle of this
Pulmonary
other
artery
model,
banding.
of another different model. Yeah,
Right.
good.
So then we went to the lab and then looked at seeing what can we actually knock down NUD21 and what happens with that.
And if NUDT1 is playing a role in the pathology of pulmonary hypertension, if it's playing some adverse effect on endothelial cells, then you might see, if you get rid of it, you might reduce it, you might see an improvement in endothelial function.
That's correct.
Okay.
That's correct. So that's what we wanted to study, and that's what indeed we found. That if you look at controls to NUD21 knockdown, you actually were able to see a lot more tubes formed, which is another assay that you can use to study the function of endothelial cells.
So these are endothelial cells growing on matrigel.
Correct.
In matrigel, they form tubes. Like trying to form a network.
Yeah. And we were able to show that if you knock down NUD21, you can increase the tube formation compared to controls, suggesting that maybe this lack of vasculature that we were seeing in RV failure was possibly affected by this NUD21 upregulation in the endothelial cells, leading to lack of microvasculature and leading to RV failure.
Yeah, interesting.
But we still, some of the things that we don't know is, of course, which pathway does NUD21 actually, affect downstream to cause this lack of, loss of microvasculature that we don't know yet, and that is something that we are doing right now to figure out.
That's exciting. You're kind of getting right at the meat of the issue. Is what's the cause of causing the vascular problem, the vascular deficiency in pulmonary hypertension that leads to right heart failure, and in fact it seems to be a problem in the right heart. This vascular problem.
Right. So, in summary, we sort of explored the expression of key APA complex in right ventricular failure, and we were able to show that there is marked increase in NUD21 in the endothelial cells from right heart failure patients as well as rats. And then we were able to look at another model in a publicly available database and show the same thing. It does look like it was, it does affect angiogenesis. And maybe that upregulation is what is driving the vascular failure. But again, I think we have a few more experiments left to be done. And again, pretty much all of. I showed Raj's photo earlier on, but I forgot to introduce him. But all of this work was done by my research scientist in the lab, Raj Amrutlingam. So
that's really exciting. So this alternative method of regulating RNA, this alternative polyadenylation, may be playing a role in the endothelial dysfunction that occurs in right heart failure and perhaps in pulmonary hypertension. We have a question from the audience. So could the loss of microvasculature in RV failure be due to a signal that detects toxicity of the content of the microvasculature towards RV myocardium or extracellular matrix? I think the question is getting at is this a response to something that's some toxin potentially?
I think it is entirely possible, and the toxin could be pressure, because what you're really sensing is the pressure overload. Now, because that's what happens in pulmonary hypertension is an RV pressure overload model. Now, whether is it the increase in pressure that these and the ensuing fibrosis which happens, whether those two are playing a role in again signaling to the endothelial cells to drive this up. I mean, it is possible, but that's a good question. I don't think we know what triggers and that leads to another question of when does this happen in the pathogenesis, right? Is this when does the switch happens where you go from a compensated to a decompensated state? And I think those are all good questions. I don't think we have the answers for it.
But clearly, changes in the environment can set up conditions for pulmonary hypertension. Also, there are genetic causes for pulmonary hypertension that may predispose one to develop.
Absolutely.
Could you talk about that?
Yeah, so I think BMPR2 mutation, which is the most common one. There are other mutations too, but BMPR, which again works on the BMP pathway, and it's very well known and which is sort of a protein in the same TGF beta superfamily. So that is well known to cause pulmonary hypertension, and that may lead to a change in the environment leading to this.
Someone else has a question about NUD-T21, and you showed that it seems to affect endothelial cells, and knocking it down enhances endothelial cell processes. Does NUD-T21 have a direct effect on the myocytes? So
that's a good question. We have not studied it, and we think, at least we've studied it in the fibroblasts, and it seems like there is this APA seems to be different in different cell types.
Sure.
And I think that's something that we are just discovering, and I'm sure Harry has some insights into it.
Well, let's turn to Harry now, since I think he's ready for his slides. Dr. Karmouty-Quintana, is it okay if I call you Harry?
Yeah, that's fine.
Well, Harry, why don't you give us some insights on right heart failure and the role of alternative mRNA splicing?
Sure. So, as you know, I already covered this areas of pulmonary hypertension. So one of the key components here is this remodeling of the vasculature, where you see this increased proliferation of the smooth muscle cells. So on the left side, you can see two vessels there with a small airway wall. On the other panel, you can see this very large vessel wall, and that leads to obstruction of the lumen that ultimately leads to right heart failure due to increased pressure on the right side of the heart due to the narrowing of the lumen. And one of the sort of in some way you can think about pulmonary hypertension and this excessive proliferation of the vasculature to have some similarities to cancer. And this is what this colleague Christophe Guignabert, who is one of the leading forces in the pulmonary hypertension field, he looked at this review looking at hallmarks of cancer and hallmarks of pulmonary arterial hypertension. And in this figure, you can see how the more red these circles are pathways that are common between cancer and pulmonary arterial hypertension, and these include mostly sort of proliferation and cellular accumulation processes, some degree of inflammation, and as Ash talked about, angiogenesis. And a lab just around the corner from ours at UT was looking at CF1M25. So here, just want to clarify with the listeners, CF25 is the same as NOTI21, which is what Ash is talking about. Unfortunately, this protein has two names, so it makes it a little bit confusing. Same thing, CF25 and NOTI21. So in this paper that was published in Nature, these colleagues of ours were able to identify alternative polyadenylation in glioblastoma tumor suppression was linked with cellular proliferation in this model. And so how this works is so a little bit about APA. I know Ash has talked about it. It's a sort of widespread mechanism that regulates gene expression by producing shorter or longer mRNA isoforms, and that's also important. And I'll have a picture on how this is represented. And as I mentioned, CF1M25, or also NOTI21, is part of a CF1M complex, and this binds to this UGUA motifs in the mRNA to mediate the cleavage reaction and assists the poly-A machinery to put the poly-A tails. What's important here is that depletion, so loss of CF25 or NOTI21, leads to widespread shortening of mRNAs, particularly the 3' UTR section. And a lot of this work was done by Scott Collum. He's now a postdoc in my lab. And this is sort of an mRNA. So you have, you have the proximal polyadenylation site. So that's where the red arrow is. And you have the coding sequence. That's where it says mRNA. And in between that, you have the 3' UTR and the distal polyadenylation site, which is in the green arrow on the other side. So typically, when the cleavage and polyadenylation machinery prefers and is much more efficient to put the poly-A tails in that distal site. Now, what happens in this complex, you can see there's many, as mentioned, there's tons of different proteins that regulate or that form part of this machinery. And CF1M25 is one of those. And many of these have been implicated to mediate changes in the 3' UTR. And what happens is that when CF25 or NOTI21 is no longer present in this machinery, then the poly-A machinery, instead of selecting the distal polyadenylation site, it preferentially binds to this proximal polyadenylation site. And as John mentioned, this can really change the sort of what we call the 3' UTR landscape in cells, because you lose a lot of regulatory elements. And these can be microRNA binding sites, can be AU binding sites. And this typically can lead to increased translation of this mRNA transcript. But one of the other things that we're also thinking about is that these microRNAs are normally able to target a normal transcript that has a well-defined 3' UTR with regulatory elements. It's now free to associate and bind to other components. So you not only have a sort of global change in the 3' UTR landscape, but you also have changes in the proteome and also the microRNA sort of regulatory environment, if you wish. And that typically happens with loss of CF25 or NOTI21. And this is how this can look like. So this is an experimental approach where we knock down CF25 in pulmonary smooth muscle cells. And as Ash mentioned, CF25 seems to have many different functions depending on what cell it's working on. And you can see in panel A, what this leads is to shorten 3' UTR. So those are the blue dots. There's much more of those. There is some lengthening. And this is done by an algorithm, which is percent of distal utilization index, which is PDUI, following CF25 knockdown. So what we did in this initial study, we looked at sort of what are the pathways that are affected. We had a list of genes and we put those into certain bioinformatic tools, and we find out that a lot of the pathways that were affected, including metabolism, extracellular matrix, or cell proliferation. Now, one of the things we like to show is in panel C, you can see this is VMA21, and we use this as a gene to test whether loss of NOTI21 leads to shortening of the 3' UTR, and these are genome browser reads for VMA21. You can see in the control, so those green, you can see a lot of peaks, but in the brown, when you have loss of NOTI21, VMA21 signals are vastly reduced, and the expression level goes up as well. This is shown also on panel D, and this is here. We are able to calculate this using RT-PCR approaches. So we develop primers that target either the coding sequence or the 3' UTR section of that transcript to demonstrate that there is indeed less utilization of the distal polyadenylation site. So in terms of pulmonary hypertension, our hypothesis was that loss of the CF25 in smooth muscle cells would induce 3' UTR landscape remodeling that would contribute to pulmonary hypertension. So this is first, this is isolated arteries from patients with pulmonary arterial hypertension, and you can see that there is a dramatic depletion of CF25. And when we looked at an animal model, and I'll show you more details on how this animal model works, we give the cells the mice hypoxia sugen for 4 weeks, and that's 10% hypoxia with sugen once weekly, which is a VEGF3 inhibitor. And after that, we can see that we have also loss of CF25 and CF68, which is another component of the polyadenylation machinery. Now trying to see whether this was consistent with the human presentation of pulmonary hypertension. These are vessels of patients with normal. This is group 1 pulmonary hypertension, so pulmonary arterial hypertension. This is group 3, which is patients that have lung fibrosis, in this case, with pulmonary hypertension. You can see that there is smooth muscle acting, which is the sort of pink-orange signals, and NOTI21 signals, which are the brown ones, and this is a nuclear protein. And if you count the amount of NOTI21 signals that are cellular, that come from, that are nuclear actually, that come from, that also express DAPI, we see that there is a reduction with patients with pulmonary hypertension, either primary, which is pulmonary arterial hypertension, or in group 3, which is IPF and PH, demonstrating that this loss of NOTI21 is consistent with the development of PH. So this is our model, where we give mice hypoxia for 10% for 4 weeks, and once a week we give this VEGF3 inhibitor that helps to make the pulmonary hypertension more dramatic, because mice are unfortunately not the best models for PH. And in this case, what we did is to try and demonstrate whether CF25 or loss of CF25 was a causative agent. We knocked it out from cells expressing transgenic-3, which targets smooth muscle cells. And we either had the mice in normal ambient air control, or mice lacking CF25, or we put them under hypoxia sugen. And in this case, what we see is that we see evidence of right ventricle systolic pressure. So this is a marker of pulmonary hypertension in mice. So we put a catheter in the right ventricle, and we can see that there is elevated levels of pulmonary hypertension. So if you look at compare the white bar and the blue bar, this is the control mice. You can see that there is a significant elevation. If you look at the gray bar, that's just the mice that lack NOTI21 under ambient air. They already have higher RVSP levels. If you put these mice in hypoxia, then this is the orange bar. You see a much greater and very dramatic increase in right ventricle systolic pressure. This is left ventricle, just to see that there's no difference. This is truly a pulmonary component. And this in panel C, you can see that there is more remodeling in the hypoxia sugen mice, and that the mice that lack NOTI21 have both remodeling and the arrow respond at Ki67, which is a marker for proliferation. So, due to some technical issues, Ash was supposed to go after, but he already gives you the overview of how this affects the RV, and as you can see, it's a little bit more complex story, where in his case there is overexpression of or increased expression of NOTI21 that seems to be pathological. So now I'd like to finish with some of the people that have helped a lot in this work. So Scott Cullum in UT Health postdoc in my lab did the majority of this work. And of course, we collaborate with Ash and members at Methodist that have done helped us a lot to get a lot of primary tissue. And we're funded by the NHLBI, NIH for an R01. And we also had a lot of funding from internal and the American Heart Association. And if anybody's interested that wants to do research in pulmonary hypertension, where we're looking at more people. And we also collaborate with other hospitals, such as Memorial Hermann, who has also an active transplant program. And if there's any more questions, happy to answer them.
Thank you, Harry, for that. There are some questions coming in, but first I'd like to congratulate you on a wonderful study, really intriguing, and congratulations also on the NIH funding. It shows that other people think it's exciting as well. One of the questions I have for you is, are there any ways that we can manipulate therapeutically the APA, this alternative polyadenylation?
That's a good question, and it's tricky because it can be a little bit of a global phenomenon, but we're working with actually folks at Methodist that one of the approaches to do some sort of nanotechnology to have a modified mRNA for NATI21 that is, does not have 3' UTR areas. So therefore, we can then rescue expression of NATI21 in the vascular smooth muscle cells. That would be one approach. Of course, I think as we have more of a opposing strategy here to try and deplete NATI21 in the RB. So that's one approach I think that could be done.
Thanks. Ash, you showed that NUDT21, this protein that's important in alternative polyadenylation, it's increased in endothelial cells in the failing right heart. Someone asked the question, does it have an effect on the endocardial cells? Or is it altered in the endocardial cells?
We have not looked at that, but the endothelial cells, I mean, the way we I guess it's a good question, but we have not looked at
that specifically.
Specifically.
You're looking at total endothelial cells, and that may have included some of the
endocardial
cells that are lining the ventricle itself. So we're still not known. How about in models of pulmonary hypertension, is polyadenylation in the left ventricle normal?
Well, that's a good question. I don't think we've looked at it. Harry, do you have any insight?
No, we have not. Well, there's a paper out there on I think circulation where they've looked at APA in left ventricle disease.
Correct.
And they found that there's evidence of APA. So I don't know if it's if they really looked at shortening of 3' UTRs, but I do recall there was APA. And there's many different other. We looked at NATI21, but there's other mediators like PABP1 that has been associated with APA.
Mm-hmm. You made the analogy between cancer and pulmonary hypertension, gene expression changes that are occurring that are common to both. Let's talk about that a little bit. What pulmonary hypertension is worse than some forms of cancer, obviously. Tell us a little bit about the similarities between the two diseases in terms of gene expression. And maybe let's focus on the endothelium. What kind of changes are occurring there?
Well, I think both in the endothelium and in the smooth muscle in PH, you have a proliferation that's somewhat uncontrolled, and there's a resistance to apoptosis, and these are sort of similar pathways that or consequences that happen in cancer. Another component that I think is interesting that links them is a context of hypoxia, where hypoxia is present both in cancer and in particularly in pulmonary hypertension. Although it's a lung, so people think that how can there be hypoxia, but indeed in PH there is, and also in lung fibrosis there are hypoxic areas of the lung, and this can drive a lot of the pathology, particularly in chronic diseases. The link with NATI21 here, or CF25, is that we've seen that at least in smooth muscle cells, that this hypoxic element can lead to a depletion of NATI21. And this could be important regulatory aspect in both PH and maybe in cancer. And Warburg effect, for example, that's studied a lot in, I think was discovered in cancer, has also been implicated in PH, where there's this metabolic dysfunction in mitochondrial deficiencies that predispose or even have been associated with the development of PH.
Mm-hmm. There are these, you were mentioning alterations in endothelial proliferation. I mean, there are these strange occlusive phenomenon in the microvasculature of the lung. Talk a little bit about that. Are those truly endothelial cells that are growing, obstructing the lumen? Endothelial cells normally form a monolayer, but in the distal vessels in pulmonary hypertension, you do see these lesions that have been thought to be due to endothelial proliferation. Are we sure those are endothelial cells?
It's a great question, and I think that's similar to a lot of different pathologies where you have this sort of abnormal proliferation or presence of cells that are accumulating, and they probably start from an endothelial component or mesenchymal, but there probably is a spectrum of, they're not clearly endothelial cells. They're developing other phenotypes, and this is, I think, in the put an analogy, I think in sort of the atherosclerosis field, where you have these smooth muscle cells that then change into some more of a macrophage type phenotype, and they begin to secrete a lot of intracellular matrix and might even proliferate more. These are, I think, similarities that might be happening in endothelial cells, where they may become acquire more of a synthetic phenotype, express more ECM components, and contribute to.
That's a really interesting idea that the endothelial cells might be undergoing some sort of malignant transdifferentiation that allows them to proliferate abnormally, because normally they form a monolayer, but you get these plexiform lesions in the pulmonary capillaries, that could be some strange cell that could be derived from endothelium. Interesting. The other thing that you mentioned that occurs in pulmonary hypertension and cancer is activation of inflammation. And there's increasing evidence that inflammatory signaling can precipitate transdifferentiation, can cause these alterations in cell fate. So that potentially that could be playing a role as well. What is the role of inflammation in pulmonary hypertension? We do have anti-inflammatory drugs. Are they useful in pulmonary hypertension?
So that's a good question. I think that is of interest, and I think there is actually some data, especially that mTOR inhibitors have been seen in small human studies to help with pulmonary hypertension, and there is actually a study ongoing with IL-6 inhibition with tocilizumab, looking at pulmonary hypertension. But I think the inflammation in PH, I think it is, and there was actually a study with Rituxan, but that was not a positive study. I think the thing with pulmonary hypertension and its role of inflammation is, where in the pathogenesis of pulmonary hypertension does it play a big role? Because the problem with PH, especially when you come to humans, is by the time they're even identified, it is 2 years on an average from the symptom onset, which means you're already in a state where you've lost most of the vasculature. So for the pulmonary, for pulmonary hypertension to happen, there is general thought process that you should lose about 50% of your microvasculature. So that's already too late. So I think a lot of the studies which are looking at inflammation are already studying it at a stage where I think by altering the inflammation, I don't think you will alter the natural history of the disease. And that's probably why they have not been very successful.
I see. So these patients are coming to us, most patients with pulmonary hypertension coming to us long after they've lost a significant part of their vasculature.
Microvasculature.
They're getting symptomatic at a late stage in the disease process. That's interesting. We know there's a question that just came up that is related to that question of can we make the diagnosis earlier? If we made the diagnosis earlier, could we have more of an effect with our therapeutics? So the question is there an imaging technique to assess loss of microvasculature in the RV, or you could say in the lung too? In patients, can we detect it earlier?
So in the lungs, so we are in our lab along with Dr. Jung Lee at the RI. We are developing a PET imaging agent, which at least in our animal studies have been shown to be able to detect pulmonary or development of pulmonary hypertension or vascular remodeling much before there is an increase in the pressure, at least in the monocrotaline animal model. And we are currently in the process of developing that clinically. So hopefully, in a year or two, I'll be able to answer that question, in humans.
That would be exciting to see a device or a diagnostic come to the clinic that could help us make the diagnosis earlier. Another question, the behavior of endothelial cells seems paradoxical in the pulmonary vasculature. Well, this is getting to the issue of the plexiform lesion, which is thought to be an endothelial proliferation that causes this late obstruction, versus in the right ventricle, it seems like you're losing microvasculature.
Yeah, I think that's and we were talking about it when Harry was presenting, that is indeed a dichotomy. And I think when we were actually studying this in the RV, we couldn't, in fact, even kind of believe the results of this, at least the APA machinery playing a role, because in one, it was being downregulated, in the other, it was being upregulated. I think
you're referring up and down. You're talking
for
about
the APA.
APA
But I see the question is, why is the behavior of endothelial cells paradoxical? I think here the insult is different, and that is what is the difference. I think in the RV, the insult for the endothelium is really the stretch and the pressure, which may be different than hypoxia, which is the primary driver in in the pulmonary vasculature, whether it's hypoxia or inflammation. So I think maybe these two stressors affect endothelial cells differently. That's again, this is a hypothesis. I don't have any explanation, but when we were thinking about why this NUD21 works differently, we were just thinking that maybe the stressor which triggers this is just different, because in RV you're looking at high pressure overload, you're looking at sort of shear, like the like increased pressure, which is different than what you see in the pulmonary vasculature. So I think that's
in one way it's the same, because in both cases you're getting a deficient functional vasculature. The vasculature isn't functional in pulmonary hypertension. It's not functional.
That is correct.
In the right heart. And aren't you losing microvasculature also in the lung and pulmonary hypertension? I mean, you do see these plexiform lesions, but you also lose microvasculature as well, don't you?
You do, but I guess you're right. You do, but you form the plexiform lesions, and then they essentially thrombose, or they. So yes, there is. The peripheral vasculature, and that's one of the ways we diagnose with, like on a VQ scan, when you see there is just loss of perfusion in the peripheries. It's just classic for pulmonary hypertension. Right. So yes, it's impaired, there, but
sites of impaired perfusion throughout the lung.
And mostly in the peripheries.
Right, in the periphery.
Yeah, because it's the small vessels which
are most affected.
Mostly affected. Harry, your thoughts?
Yeah, I think. Try to also try and understand why there is this sort of paradoxical effect on the endothelium and the smooth muscle. And if I recall, when we do our staining for NUD21, it seems that the endothelium may pick up more NUD21 signals. And understanding that is, I think, a little bit more pretty not as straightforward, let's say. And I was thinking that perhaps the stretch may be one of the mechanical component there, could be one of the things that is different compared to the lung vasculature, that may lead to a overexpression of NUD21 compared to the lung smooth muscle. Another component, I think that so I think in PH you do have pruning of the vascular tree, but at the same time you also have a proliferation of that remaining component. So it's a pretty dynamic response.
Well, I want to thank you both, because I learned a lot with your presentations today, and I think one of the key things that we learned is that RNA is dysregulated in pulmonary hypertension and right heart failure. That's one important lesson. That dysregulation is due to alternative polyadenylation, which can change the message RNA, make the UTR longer or shorter, and that can affect the regulation, the stability of that RNA, and thereby affect the proteins. And then the other thing we learned today is that right heart failure is a form of vascular failure. Pulmonary hypertension, obviously, is a form of vascular failure. So I want to thank you both, gentlemen, for shedding some new insights into pulmonary hypertension and right heart failure. Dr. Ashrith Guha at Houston Methodist Hospital and Dr. Harry Karmouty-Quintana at UT Health. Thank you both for joining us.
Thank you very much, Dr. Cooke. It was a great pleasure.
And thank you all for joining us at The Cutting Edge today.